Any questions before we get started? So today, I'm gonna finish talking about sequence alignment and finish up this seq_analysis.r script. Right now, I am at line 229 of the .r file, and we talked last time about if we have two DNA sequences and we want to align them together, pairwise alignment of DNA sequences, we can use the rules of match reward, mismatch penalty, and gap penalties that we select to find an alignment or alignments that achieve the best possible alignment score, like in those PDF slides. I was going to show you today what happens if you want to align protein sequences instead. Mentioned, I think, just briefly last time that it's a little more complicated when you get to proteins, because now you have, instead of 4 bases that can line up or not line up, you have a bunch of amino acids that can line up or not line up. And as we talked last time, there are different combinations of possible pairings of amino acids that are either more or less likely than others. So the penalties and rewards that are typically used when aligning amino acid sequences are more complicated than what we were using for DNA sequences. So I have libraried in biostrings, and we're going to align a couple of amino acid sequences. So down here, 2 toy sequences. And we're going to select a set of rewards and penalties that are commonly used, or that is commonly used. There are multiple ones. And if I say data, tell me the data sets that are part of package biostrings. Here's what I get. So each of these BLOSSOM items and these PAM items are scoring matrices. We're going to use one of them. I'll show you what it looks like. We are going to use the BLOSSOM 62 score matrix, and looks like this. So these are rewards for putting, say, an A together with an A. We're going to get a reward of +4. If we were to put an A together with an R, we'd get a penalty because it's a mismatch, and that particular pairing is given a penalty of -1. Whereas an A with an N is given a penalty of -2. So these are all chosen based on expert knowledge of the chemical structure of these amino acids. Choosing between, should I use, say, BLOSSOM 62 or PAM 250, what set of penalties and rewards is most appropriate for me? Depends on the setting, like what exactly you're studying, what kinds of misalignments you want to especially guard against, for example. For our purposes, we're just going to illustrate doing this with one of them, the BLOSSOM 62. All right, so I'm going to create an object called myScoring Matrix, and it is just the name BLOSSOM 62 that I'm going to pass to a function in a moment. Okay, here's our 2 sequences, and we're going to pass them as before to the pairwise alignment function. The same arguments are applicable. There's a substitution matrix, which we used that nucleotide substitution matrix function before to do DNA sequences. There's gap opening and there's gap extension penalties. So the substitution matrix is that BLOSUM matrix. It's what happens for matches and mismatches. But otherwise, this function looks just the way it did before. Yes? So these two sequences I made up. If you had a long sequence and wanted to pull out a subset of it, let's make these over in R real quick. So seq2 is that. Suppose I wanted to pull out the subsequence that lives between, that includes the 4th, 5th, and 6th, and 7th letters only. Like I want to go to chromosome 1 and get bases 1,012 up to 1,420. I want to pull those out and only consider those. You can manipulate characters and strings with a couple of different functions. There is a substring function, and if I say give me a portion, only a portion of this particular string, I can say start with character 4 and end with character 7. And it will only pull out the corresponding subset of this bigger sequence. So you can extract smaller pieces to work with. Once we have a couple of sequences, let's see what happens when we try to align. We are going to do a global alignment. I'll show you local alignment in a second, because we've said they can be different, and they are different in this example. Here's global alignment. Gap open. Okay, I didn't define gap open yet. All right. Thanks. So that was the alignment, and the score was -19. We could match that score if we looked at that BLOSUM 62 scoring matrix and go through and say, okay, there's a P with an E, what's the penalty for that? There's an A with an A, what's the reward? W with G, what's the penalty? Continue, and then here's a gap of length 1, what's the penalty for that? Add it all up, it comes out to -19. Okay. Let's do it again, and let's try a local alignment. See what difference, if any, we get. That's what the local alignment comes out with, and the score 18 in this case. And notice that we don't have all of the two sequences printed out here. We only have taken this A, W, H, E, and found a good local match for that portion over here, specifically the latter portion of the second sequence. So all I'm shown is the letters, the bases, or the amino acids that were actually lined up with amino acids from the other sequence. So the stuff that's not shown was basically ignored for the purposes of this alignment task. That's an illustration of global versus local alignment. All right, here's a picture. You saw this picture once, but I'm going to show you how to make it. This is a picture that kind of qualitatively shows you visually how well two sequences align. And let's make one and then talk about it. So we're going to read in the sequences for, I think, a gene that we've seen before, RPOB, and we're going to read it in from two different species, M. tuberculosis and E. coli. We've done this before. So let's read these in. Going to GenBank again. Query to use seq_in_r to download sequences. Okay. And then I need to make sure this is like it was above. We're going to pull out, there's multiple sequences that came out because there's multiple protein products for each of these genes. And I'm going to pick one representative of each from each species to compare. And we did this earlier, 649 and 1. So I'm going to change this 646 to 649 so that I'm comparing the same two sequences that we did before. Use the getSequence function to extract the actual sequence. So now we have our POB tuberculosis sequence, and this something similar for E. coli. Now we're ready to make this picture, and there's a built-in function that does it for us. I think it's a seq_in_r function. So this function comes from the seq_in_r package. And let's see. Most likely the oldest visual representation used to compare two sequences. What does it look like? I'm only going to look at the first 150 bases of each because we're going to put a dot, we're going to make an array of dots in a scatter plot with horizontal tick marks for every base in the first sequence and vertical tick marks for every base in the second sequence. So if there's lots and lots of bases, it's going to be a very busy plot. To illustrate it, we'll just take the first 150 bases. All right. Here is a dot plot for these two sequences. The representative from M. tuberculosis is on the horizontal axis, and that for E. coli is on the vertical axis. So there's 150, imagine, tick marks on the horizontal axis. There's 150 on the vertical axis. And if you go to any one location on the horizontal axis, like let's go out to 20. At that particular spot, if you look up, through the column sitting on top of 20, you're asking for the 20th base of M. tuberculosis. What is that base? Might as well look at it. RPOB seq 20 is a T. Now go through all 150 bases in the E. coli sequence, and is each base a T? Yes or no? If it's a T, put a red dot in this picture. So above 20, there's a red dot just a few bases in. So if I look at RPOB E. coli, first 10 bases, I guess it was the second one. The second base is a T. 5, 6, 7th bases are all T's. So there's the second base, and then that little block right there is the 5th, 6th, and 7th bases. Everywhere there's a red dot above 20 means E. coli's base matches this 20th base. And a white dot, if there's not a dot, basically, that means it does not match. Okay, so suppose, what does this picture tell me then? What would be indicative of a strong alignment would be if there were diagonal stretches of continuous line segments. A diagonal stretch, like here, if you look in that region right there, there is a diagonal stretch going like this for a little ways, which indicates that for this collection of bases in M. tuberculosis, there is a match throughout a corresponding region in E. coli. So diagonal means this T matches with that T, and this A matches with that A, and so on, for a contiguous stretch. If they matched really well, you would expect to see several diagonal stretches. If they were perfect, if they were the exact same thing, I'll show you a picture in a second, you would see an exact diagonal line across the entire picture. All right, here's what it looks like if they are the same. I'm just going to do the same exact code, make a dot plot, but notice that I'm doing M. tuberculosis both times. So make a dot plot of the MT sequence against the MT sequence. And it looks like that. So I guess I'm looking for diagonal going that way. Either way actually is indicative of alignment. So lots of red and white everywhere, but up this diagonal there are dots throughout, indicating that that sequence lines up perfectly with the other sequence. Okay. So you would get practice if you were doing lots of alignments and making pictures like this in looking at one and saying, no, these sequences have nothing, they have nothing in common. Or, yeah, these sequences have a lot of overlap. It's a qualitative tool. For the purposes of doing a formal alignment, you would like to have the algorithms that we've already met that formalize things, and you would like to be able to quantify your confidence level in an alignment. And I'm going to show you how to do that in just a second. So let's talk about that. Suppose we do an alignment of two sequences to each other. Let's say we did pairwise alignment. How confident are we? Because we said before the algorithm is going to give us an alignment every time. The best alignment is such and such. Here's my score. But that on its own did not tell us whether it was an alignment that is believable or just the best among a choice, the best choice among a bunch of bad ones. So just like in statistical inference, you like a p-value or a confidence interval because they give you a way to quantify how confident are you in your conclusions. We can't guarantee that the mean survival time with treatment is necessarily bigger than the mean survival time on control because we only have a sample of 200 people to study. But if I can say I'm 95% confident that the mean difference is between 1 and 1.5, that's valuable, information. So here, two sequences that we've aligned somehow. Is it a better alignment than you could expect to happen by chance, is the question we're going to ask. If we had two sequences that, let's say, were of the same length as these two and made up of the same collection of amino acids. So we're going to pick two sequences, they're that length, and we're going to populate them with these possible amino acids. Suppose you just randomly made up two sequences with amino acid letters and did an alignment of those two. That alignment is a bad alignment because those are completely randomly made sequences. They do not line up. They have nothing in common. I can still ask the alignment algorithm to do its best and return to me its alignment and its alignment score. Okay, so what do we do when we do a hypothesis test, by the way? Let's go back to a familiar example like a t-test. We have some people with under a treatment. We have some people who are in a placebo group, say. And after our study, we compare some response variable like survival time, and we want to make an assessment, a test, make a conclusion. Is the mean survival time for treatment different than that for control? The way that we do that, that you end up looking at a p-value or a confidence interval. But let's think about the p-value. What does the p-value represent, and how do you compute it? The p-value is the probability that if your null hypothesis is true, you would see as much evidence that the null is not true, or even more, just by chance. So in the treatment versus placebo example, let's say we look at survival time, and people in treatment, on average, their average survival time was 2 months longer than the placebo group. That 2 months, that number, is a statistic. It's called a statistic. It's a number you compute from sample data. Because your sample is randomly obtained, your 10 treated individuals and 10 placebo individuals were randomly obtained from the whole population of people with whatever disease you're studying, say. So the data that you see, those 20 numbers, were randomly obtained. If you went and got another 20 people and randomized them to treatment or placebo, you'd see 20 different numbers probably. And so you would probably see different averages for the two groups and a different mean difference. So if when my 20 numbers, my sample, I saw a mean difference of 0.5 years or something, the statistical question, the inferential question would be, is 0.5, a difference of 0.5 observed on 20 people, is that enough, is that far enough away from zero for me to be confident that the real population level mean difference is not zero? Because suppose there is no difference between treatment and control. There's nothing, you just give, your treatment is not good, it doesn't do anything. If you randomly picked 20 people from the population of all people, and you randomize half to get the treatment, half to get the placebo, just by chance, you might have gotten healthier people in the treatment group, just by chance, and less healthy people in the placebo group. In which case, just by chance, you would get a mean difference of maybe 0.5 or even more. But it's not a real difference. It's just random variability. So the p-value is about, suppose that there is no difference between treatment and placebo, how likely would it be if we randomly drew people from that population to see a mean difference of 0.5 or more? That's what the p-value is. It is a probability. It's the probability of seeing as much evidence as we saw, or even more, against the null hypothesis when it's really true, which means it just happened by chance. So we're going to do an alignment, and we're going to get some score, like minus 19 is the score. We can ask a similar question if we have two sequences made up of this many letters, and these letters are drawn from a pool of 20 letters, say. If you just randomly constructed sequences and started aligning them, what proportion of the time would you get an alignment score that high or even higher? If the proportion of times that you got an alignment score that high or even higher was really small, that would indicate that you would not expect to see that strong of an alignment by chance. Suppose the proportion of times that randomly generated sequences that are like these have a score of minus 19 or bigger. Suppose that that's 0.01, the proportion of times. You compute that somehow. I'll show you how to compute it in a second. Yeah, it's a lot like a permutation test is what we're going to talk about. A permutation test is a way to simulate, suppose there really is no difference between my comparison groups, say. It's a way to enforce that the null hypothesis is true. And a p-value is the probability of this much evidence or more if the null hypothesis is true. So if you can find a way to force the null hypothesis to be true and then kind of simulate, you can just look at what statistics come out and ask, are they unusual, or is my observed one unusual relative to them? If it looks just like them, then probably it was generated randomly, like they were, and probably the null hypothesis is true. But if it stands out relative to them, then this does not look like them. This looks like maybe it was a real alignment. All right, so let's continue here going down. Here's what we're going to do. We're going to have two sequences, and one of them is going to be shorter than or equal to in length to the other one without loss of generality. So take the smaller one. Here's one way to do this. Take the smaller of the two and randomly generate a sequence that's just like it. Randomly generate a sequence of letters that is the same length as the one we're about to play with, and pick letters to start stringing together to be comparable to the proportions in the original sequence. So here's my two sequences. I have a really long DNA sequence, and then I have an even longer DNA sequence. I want to align the two. Take the first one that's shorter than the second one, or it could be the same length as the second one, but not longer, and tally up what proportion of A's, what proportion of T's, what proportion of C's and G's. So it's 21% A's, 19% T's, and so on. Now, many times, capital B times, which I'll take to be 1,000 or something, randomly construct a new sequence by going, it's going to be the same length as my first sequence, and go to base 1. What letter do I put in base 1 in this simulated version? Well, draw an A with probability 0.21. Draw a T with probability 0.19, draw a C with probability whatever its proportion was, and so on. And put that in position 1. Then go to position 2 and do the exact same thing. Give me a T with probability 0.21, a C with a probability 0.19, and so on. Dot, dot, dot. Now you have a completely random sequence of the same length as the one we started with, and it has a similar composition to the one we started with. It's got the same proportions on average of the different letters. Now align that to the second sequence and record the score, the alignment score. After doing that many times, each time we create a random version of the first sequence. When we created a random version of the first sequence, we kind of forced the null hypothesis to be true of a false alignment, because the randomly generated sequence has nothing at all to do with the second sequence. They should not align because the first one was just randomly made up. So every time we do this, capital B times, we're going to get an alignment score. The best alignment I can do of your randomly generated sequence to the second one that we're comparing to is such and such. And I'll have 1,000 of those. So then we could take like our minus 19 that we had up here and say, that's what I observed. That's the data that I have. Here's my simulated versions of this statistic. That's a statistic also, minus 19. It's a number computed from data. So it's random. Every time you sample sequences and do this, you're going to get a different one. I observed a statistic of minus 19. My simulated versions under the null hypothesis of a completely random alignment look like such and such. Maybe draw a histogram of them. In fact, let's draw a histogram. Here's my 1,000 alignment scores that were generated by randomly create a sequence and then align and record the score. These are what you would expect to see just by chance. Some of the time, you're going to get solid alignment scores just by chance. Our alignment score, I don't know where it fits on this exactly, but let's say it's down here, minus 19. That's what we observed. How likely is it to see an alignment score of minus 19 or stronger just by chance? We could answer by computing basically the area under the curve to the right of my observed score. That's an estimate of a p-value. The probability that just by chance, they shouldn't align, but just by chance they do align with a score like ours or even stronger. So if the picture does look like this, the area under the curve, total area under one of these types of curves is 1, because we're treating this like we're talking about probability. So the area under the shaded area under this curve will be less than 1. In this case, it's going to be kind of close to 1 because we've shaded in most of the picture. So the p-value is going to be big. Typically, we would say p-value less than something small like 0.05, 0.01 is small. So if I had seen an alignment out here of 15 or something, the p-value, the area under the curve to the right of it, would be very small. Okay, so our p-value is going to be the proportion of these capital B simulated sequences that had alignment scores as big or bigger than the one that we saw originally. And if that p-value comes out small, that indicates a strong alignment. You can be confident that this alignment resulted from two sequences that really do line up with each other. So I have two functions that I've written. In R, you can create your own function pretty easily. Like if I wanted a function that takes as input a vector and adds 1 to every element of the vector. I could say function is, or f is a function of an argument that I'm going to call x. And what I want to happen inside of this function is whatever x is, whatever's passed in as vector x, save as output x plus 1, and then return to me the result. So f is now a function. It's an object of class function. And I can call that function by saying f of and passing in a vector. So let's pass in a vector 1 to 5, say. And it spits out my argument. I'm going to create two functions because I don't know of a built-in function that does what we're about to do. The main function is here, and this is going to compute a p-value for an alignment. You provide the first and second sequence. You provide one of those substitution matrices, what happens for matches and mismatches. You provide gap open and gap extend penalties, and you provide how many simulations do you want to do. That capital B is analogous to how many permutations do you want to do in a permutation test, or how many bootstraps do you want to do in a bootstrap, if you're familiar with the bootstrap. Inside here, we are going to do a couple of things. First, we are going to compute the alignment score for the two sequences that were passed in. So you take the original two sequences and you compute pairwise alignment and record the score. That's your observed statistic value. If we were doing a t-test, I would be writing down my t-statistic, maybe. Now what we need is simulated versions of that score where the simulation is done in such a way that we force it to be a completely random alignment. So I have a separate function for creating a bunch of random versions of one of the sequence that was passed in, the first sequence that was passed in. I'll show you that function in just a second. Once you have that, then it's a loop. Loop over all the simulations that you want to do and just apply the pairwise alignment function again. But I'm going to pass in the ith random sequence that I created. So now align just a random collection of letters to the second sequence and store the alignment score. So right here I created a vector of length capital B, just empty placeholder vector. Inside the loop I'm writing down the alignment score for each time. then the p-value is the proportion of times that those simulated alignment scores were bigger than or equal to the one that we originally observed. If I do this inside the mean function here, this is going to take element by element, all capital B simulated alignment scores, and compare them to our original alignment score. This is just one number. And it's going to give me a vector of trues and falses. True, the first simulated alignment score was bigger than or equal to the one you saw. False, the second one was not bigger than or equal to. True, false, 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 dot, dot, dot. Trues and falses by R are treated like zeros and ones. So if you I think if I do false plus true divided by 2, yeah, it treats false like 0 and it treats true like 1. If you do something that involves arithmetic on an object that is trues and falses, it will treat them like zeros and ones. So if I say, what is the mean of this vector of trues and falses, it's going to add up how many ones there are and divide by the total number. It's the proportion of times that the simulated score was bigger than or equal to the observed one. And that is what we're calling the p-value. Okay, let me show you this function briefly, and then we'll run it and see what happens. This function, you pass in the first of your two sequences, the one that we're assuming is less than or equal to in length, and you pass in how many simulated versions do you want. And here's what happens. We're going to do a little bit of preliminary stuff, nothing terribly important there. I'm just reformatting the nature of the way the sequence is written. Then I'm going to tabulate how many times out of the total length is there an A. How many T's, how many C's, how many G's. And then compute the proportions that those correspond to. What proportions are there for A and for T and C and G? Those are going to add up to 1, but now I have numbers that I can use to generate. I don't want, like if my sequence is lots of A's and only a couple of T's and half G's and C's or something, and I'm going to simulate kind of a randomized version of that sequence, I would like to have same rough proportions in the simulated one. Just makes it more comparable. So that's what I'm doing here, is making sure that I represent the A's, C's, T's, and G's with a similar frequency. Then we're ready to actually generate the random sequences, and I do that in a loop. What happens in the guts here is that we sample. So sample, if X is the vector 1 to 5, if I do sample X, it randomly scrambles the values of X. If I did it again, I'd get a different scrambling, and so on. There is an argument to sample. Remember that if you do question mark function name and you get something like this, that means there's multiple functions named sample floating around. In the parentheses underneath each of these entries, it tells you which package the documentation corresponds to. This stuff came from either seq in R or biostrings. Base is what comes in to R when you install it. And sample is a built-in base function. So I'm going to go here. Here's the documentation. You specify what you want to sample. You can specify how many items you want to resample from it. Like if I were to say X only give me 2, it's going to randomly pick 2 of the 5 to give me. You can also say whether to sample with replacement or not. By default, it does not sample with replacement. And you can specify this, which is a vector of probability weights for each of the elements that are going to be sampled. In our case, we have A's, C's, T's, and G's, and I want to give each of those their own proportions that are analogous to what were in the original sequence. That's where I'm going to pass those proportions that I computed above. So we're going to sample seq letters I got from up here. I just pulled out the letters that were actually in my sequence. Take a random sample with replacement from those letters and use these proportions as probability weights. Here's what happens. Let's run this code. So let me run these functions, define them. Right. Here are the two sequences that we're going to play with. Here are the reward/penalty details. Let's just apply gen random sequences real quick to see what it does. All I need to do is pass the first sequence and tell it how many I want. So let's do random seqs is gen random seqs seq1, and let's just do 10 to illustrate. Random seq now contains that. Those are randomly generated kind of analogs of that guy. The E's and H's and P's and W's and all that were chosen with probabilities based on the frequency with which they were seen in the original sequence. Yes? So we are sampling with replacement here because, for example, my sequence has repeats of the same letter. There's multiple E's, there's multiple A's. So yes, I'm going to sample with replacement from these letters, and I'm going to give each one of these unique letters the probability of being selected that is the proportion of times that it showed up in the original sequence. So it is sampling with replacement from these letters. So if we had DNA with just 4 letters, I'd have lots of A's, C's, T's, and G's. I would compute what proportion of those letters are A's, what proportions are T's, and so on. And I would create a sequence the same length as my original one, and each base would be generated according to those frequencies, those proportions that were seen in the original one. So I should end up with roughly the same proportion of A's, C's, T's, and G's in my sequence, regardless of whether it's A, C, T, and G, or like in this case, these amino acid codes. We're going to take what letters are there in the sequence and randomly generate versions. How's that? Okay? All right. So let's apply it. We did this sequence alignment above, and we did the global alignment. We did both global and local. We're going to do a global alignment. So my function, I should probably add another argument to this function that would allow me to specify global or local. That would be easy to do. I would add an argument, align type, and then I would have to specify what that is here. So I won't do that right now. Maybe I'll change it for next time. Right now, this is kind of hard-coded to be for global alignment. Right. So we did the global alignment of these two sequences earlier, and we saw a score of -19, I think. So global alignment of, I think, the same two sequences. gave us a score of minus 19. Same, same. So is minus 19 good or bad? Is it a good alignment or is it a bad alignment? Let's put a p-value on it. So we are going to do align p-value is the function we just defined, sequence 1, sequence 2, gaps and penalty stuff, and let's simulate, let's create 1,000 randomized versions of the first sequence. Might take a moment to run because you're doing a loop of 1,000 things, and every time you're calling pairwise alignment. All right, so if we look back up here at the definition of align p-val, here I know what it's going to give me because we wrote the function. Here's what it's going to return. It's going to give me a list with 3 elements in it. The first element, TT0, is the observed alignment score when we just did the alignment of those 2 sequences to begin with. The second entry is going to be all 1,000 of the randomized based alignment scores. And the third is the p-value. So, names out, those are the things that are available to me. What's the class of out? It is a list. What's the length of out? It's length 3, but each element is its own thing. Out $TT0 was the original score. Length out $TTB, 1,000 alignment scores in there. And then the p-val is there, 0.863. So let's look at the first few randomization-based alignment scores. Out $TTB, just show me the first 25 of them. They look like that. So these are all optimal alignments for a randomly generated version of our first sequence to our second sequence. First time, even though they have nothing at all in common, we had an alignment score of minus 17, which was greater than what we saw the first time. The second time was minus 10. The fourth time it was really bad, minus 26. But these are all randomly bobbing around. So if I made a histogram of these, here's how I can make a histogram. Pull out the second element of my output object, because that contains those 1,000 alignment scores. And let's make a histogram. Looks like that. So thinking back to this picture, the p-value that I want, it actually looks kind of like that picture. I'm going to come out to minus 19 and ask what proportion of these observations are to the right of minus 19. I can add a line at minus 19 to make it visual. So shade in all of that stuff to the right. What proportion of the whole is that? That is the 0.863. About 85% of all these randomization-based alignment scores were better than or as good as or better than the one we saw. So if somebody gave you a p-value of 0.863, how confident would you feel? So in this case, we did an alignment of 2 sequences to each other. We got a p-value of 0.863. Was it a good alignment? Not a good alignment. You'd expect that quality of an alignment almost every time just by chance. I mean, it's not completely consistent with these 2 sequences are totally random relative to each other. Yes? Mm-hmm. So for the homework, where you are asked to do this, is it number 4? So in part B, you're going to use, or in number 4, part B, you're going to use DNA sequences to do the alignment. What we just did was for amino acid sequences. When you want to align DNA sequences, You still need these pieces because we're still going to use pairwise alignment. These are provided in the homework. The substitution matrix, you can reference some of the earlier code. So I'll show you where. Nucleotide substitution matrix. You can just create one. If you say what the match reward is and what the mismatch penalty is, there's only 4 letters, and assuming you want match of 2 for every match and mismatch of -1 for every possible mismatch, this will give you that 4x4 matrix for use. You can use this to create your scoring matrix, yes. Okay, so this was a poor alignment, and the p-value helps us to know that. So if we were doing sequence alignment, like in a large scale, maybe a next generation sequencing application, you have millions of these short little reads, and you have that whole mouse genome that you want to map everybody back to. You're going to do millions of alignments. Each time you do an alignment using this method or something analogous, hopefully in the data that you get back, you have some kind of confidence score for each alignment, like a p-value. And then you might filter. Here's my millions of alignments to the mouse genome, but some of them I don't believe are real because they had poor alignment quality. So let's filter out to only keep the reads that align to the reference with a p-value of less than something. You now have a quantitative way to say, is this alignment good or bad? We're basically done, so one 30-second blurb, where we're going to talk soon about a statistical issue, multiple testing, which is relevant to what I was just describing. So if you had millions of reads, and for each read you did a little test, like we just did. Is this alignment to this part of the genome a good alignment or not? P-value. Now let's go to the second read and do the same thing. P-value, 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 millions of p-values. We have to keep in mind that when we do a test, there's a chance we'll make an error. Even if I get a p-value that's 0.00001, that can happen just by chance. And when we do classical hypothesis testing, we say, I made a decision, the treatment works relative to placebo, and I did it at a type 1 error rate of such and such. Or I'm 95% confident. That type of language. That type of language is geared to work when you're doing a single experiment and a single test. Or at least one test for every distinct scientific question in your study. As opposed to, here's an enormous data set, let's just start doing tests and looking for stuff. If you open up any data set, maybe just generate a random data set full of all random numbers. If you start making every comparison possible, this column versus that column, this subgroup versus that subgroup, you're going to find something statistically significant eventually. Type 1 errors happen. If I do millions of hypothesis tests, and each time I get a p-value, let's say we filter our reads based on whether they had a p-value less than 0.05 or not. I'm only going to keep the reads that align with a p-value less than 0.05. That 0.05 is misleading now. You're not really 95% confident in the whole alignment because every time you did it, you had a 0.05 chance of making an error. So overall, how good is the alignment? It's not 95% confident. You got to kind of incorporate all those tests that you did at once. So I'll talk to you about something called the false discovery rate that is used as an alternative measure for errors, and it's more applicable to bioinformatics and high throughput type things where you're often doing lots and lots of tests. We'll get to that next week or probably the week after. Okay, questions? I'll see you on Wednesday. Go ahead and use the same, so the gap, this stuff here for gap penalties and everything, use that throughout. Yep. Yes. Is it this Friday? I think so. Yeah, the 12th. And I'm on the discussion board all the time, so if you have questions, just holler on there and I'll help quickly.